Just a second.
Yes. Marco is a research associate at UBC Vancouver with the Lauren Riesberg Lab. And today he's going to talk a little bit about the UV patterning on wild sunflowers and gene function of those. So go for it, Marco.
Thank you. Thank you, Nikki. And I also want to thank Olivier and Nicolas for inviting me to give this presentation. It's really great to be here. As Nikki was saying, today I will talk a bit about our work on wild sunflower adaptation, and in particular on patterns of flora UV patterns in wild sunflowers. Yeah. So as you know, in nature there's a lot of diversity, not only between species but also within species, meaning that there are populations within a species that can have very different characteristics. And we like to think that at least part of this diversity has some adaptive value, meaning that it improves the chances of survival of those individuals in the environment in which they grow. And I think there's a lot of value in studying this adaptive phenotypic variation and trying to understand what are the genetic factors that control it. And this is partially because it allows us to understand better what are the evolutionary mechanisms and the evolutionary mechanism, yeah, and the I want to say something that are anyway. So the way in which this this adaptive variation evolved and this control, and what are the forces that controls the evolution of this variation, but also it. Sorry, just give me a second. Yeah, but also it can provide us information about what are the genetic patterns that control this variation, and if we understand what are the genes that control this variation, we can also know something else about the way this gene work, what is their function, and how is this function modulated in natural populations. I don't have hopefully to convince you that wild sunflowers are sunflowers in general are a great model, and wild sunflowers are a great model for studying adaptation. And this is because there's many species of wild sunflowers. They are all native to North America. They are mostly interfertile, so you can do all kinds of crosses if you want. And most importantly, for what is this talk is about, they are adapted to grow in a variety of different environments. So they grow on sand dunes. You see some examples here on the background. Just on the sea, they grow in very dry places, desert. So there's a lot of raw material for studies of adaptation in these plants. And also there's extensive genomic resources. So we have now several reference genome assemblies. We have assessed thousands of accessions we can get and look at for wild sunflowers, and many of these have been also resequenced. And in particular for sunflower, as you know, wild sunflowers have been used extensively in the past in breeding programs. So there's also a chance that if we understand how these plants are able to survive in a particular environment, there might be a direct application for this information. So we can use alleles that we find in the wild for improvement of the sunflower, of cultivated sunflower. One trait that is almost stereotypically adaptive in plants is flower color. Flower color and patterns and other flower characteristics are mostly driven. So the evolution is mostly driven by the coevolution between plants or their pollinators. And obviously, there is a lot of diversity for flower color between species, and there's often a lot of variation also within species. And then you have some flowers, and some flowers are just yellow. And here, of course, I'm referring to the color of the ray flowers of the legals. And indeed, to the human eye, all sunflowers, if you exclude a few ornamental cultivars, are just yellow. But if you were a bee, things would be different. Bees have photoreceptors that can perceive the near ultraviolet radiation, which is the case also for many other pollinators. And so they would see something very different in the sunflowers. The pattern here that you see on the left is the most common in sunflowers and other plants. You have accumulation of UV absorbing pigment at the base of the legals or of the petals, depending on the flower. That form, and then the rest of the legal instead is UV reflective, and so across the whole inflorescence it forms a UV bullseye. And the reason why these UV patterns are there is that the pollinators really like them. People have been doing experiments on UV patterns for a long time, doing all kinds of stuff like cutting and pasting different parts of the petal to form different patterns, using sunscreen to alter these patterns. And in all cases, what they found is that when you have these UV patterns, the flower attractiveness is increased, so you have more pollinator visits. And in sunflowers is radially symmetric, of course, so it's easy to measure the size of this UV bullseye just as what we call legal UV proportion, which is just the length of the legal, so the length of the part of the legal that is UV absorbent divided by the full length of the legal. And UV patterns are common in many species, and they seem to be ubiquitous in sunflower. This is work that we did with Srinidhi Olalu and Ben Blackman. We look at 18 species of subspecies of wild sunflowers, and as you can see, all of them have at least some level of UV patterns in their inflorescences, and you see them here projected on this tree of sunflower species. The black dot here on the side is proportional to the size of the UV patterns. So you can see that they are everywhere in all the species that we checked. There's a lot of variation between species. And we wonder if there is a lot of variation between species, could there be also variation within species for this trait? Fortunately, a few years back, we started a large-scale survey of phenotypic and genetic variation in wild sunflowers. This all started with Dylan, a former postdoc in the lab, so he drove a ridiculous amount of miles across North America and collecting seeds and soil also from seeds from thousands of plants from hundreds of wild sunflower populations. He focused on 3 species of wild sunflower. This is the first one is Elianthusannus, so the common sunflower, which is the progenitor of cultivated sunflower, of course, and that's what I've been mostly talking about today. He also collected a lot of plants for Elianthus petiolarius, which is the prairie sunflower. Like Elianthusannus, there's a very broad distribution in North America, but it prefers sandier soils. And then he also collected Elianthus algophilus, although I just briefly mentioned this later on, which is the sister species of Elianthusannus, and it's endemic to southern Texas. So he brought all these seeds back, and then we grew 10 plants from each of 150 of these populations in a common garden here in Vancouver. I don't know how many of you are familiar with or have grown wild sunflowers before. They tend to be fairly tame when they are small, but then they grow to be enormous and very branched. So this is a picture at the beginning of the field season, as here that is toward the end of summer. So you see on the background, you have this wild annuals plants that are about 4 meters tall. Here you have a teddy bear sunflower and then Natalia Berkovich for scale. So they everything gets very messy. Nonetheless, we managed to phenotype these plants for a variety of different traits across the whole season, and then we collect the pictures and analyze different traits afterwards. And one of the traits was UV pattern. So in between this field trial, this common garden and another common garden experiment we did a couple of years back, we had information for many hundred different plants, especially for Annus and Petiolaris. And what we found is that there is extreme variation for this trait within species, especially in Annus. So you see there is everything from plants that have legals that are almost completely UV reflective to plants that have legals that are completely UV absorbent. So when we saw this, it's all this is very interesting. And we had basically 2 big questions, right? The first is how this variation achieved. So what are the genetic and metabolic pathways that control this variation in wild sunflowers? And why is there such variation? Is there any factor? Is there a factor, if any, that controls how this variation is distributed across the range of wild annuals and wild sunflowers? So we started with the first question, and we have the fairly straightforward way to try to answer it. Because we not only grew these 1,500 plants in the common garden that I showed you before, but we also resequenced all of them. As you know, sunflowers have fairly large genomes. So these species in particular range between 3.2 and 4.5 gigabase per. So it took a lot of sequence it's like a large sequencing effort to be able to sequence them all. And this is really thanks to Greg and JS that wrangled with all this data and managed to make sense of them. We had, while sunflowers are hugely diverse, and not only phenotypically, but also genetically, we had hundreds of millions of polymorphisms within these species, and we kind of boiled them down to sets of 4 million very high quality SNPs. And here you see for each species, and here you see a tree based on part of this data. And this tree looks like it should look, fortunately. So you have Lianthus annus up here with cultivated sunflower nested in. Argophilus is just down here, so it's the sister species. And down here you have the two subspecies of Lianthus petiolaris, so Petiolaris petiolaris and Petiolaris fallens. So now we have genetic data for all these plants. We have phenotypic data for most of these plants. So we put everything together and did some genome-wide spatial mapping experiments. For Petiolaris, so as you've seen before, the two subspecies are fairly different. So we had to analyze them separately. We found no significant association with UV pattern, floral UV patterns for Petiolaris fallax, but for Petiolaris petiolaris, we found a few significant associations, which is what you would expect for a quantitative trait. So that's nice. But then for annus, we found something much nicer. We found one locus that explains basically 2/3 of the variation that we observed within this population. And when we went and looked a bit more closely what is in this region, we found that the SNP that had the strongest association with floral UV pattern is in the promoter region of MIB111. So it's just about 5 kb upstream of this gene. And this SNP has a very strong effect on floral UV pattern. So you have plants that are homozygous for the small allele at this SNP have about 1/3 of the legal that is UV absorbent. Plants that are homozygous for the large allele of this SNP have about 3/4 of the legal that is UV absorbent. And heterozygous plants have an intermediate phenotype. And this actually, like this very additive phenotype explains quite nicely the sort of trimodal distribution of variation that we have for UV patterns in annus. So what is MIB111? So the sunflower homologue of MIB111 is a member of a small family of transcription factors. So there are 3 transcription factors that are also called production of flavonoglycosides. And as the name implies, they're involved in controlling the transcription of genes that are involved in the production of flavonoglycosides. Flavonoglycosides are a type of flavonoids. They have been involved in responses to a number of different biotic and abiotic stresses, like cold, ultraviolet radiation, and drought. And also they are the UV absorbing pigment in a number of different species. So they make for a very good candidate as a UV pigment for sunflower as well. And indeed, when we check sunflower legals, so we found that in legals that are highly UV absorbent, so they have these large UV patterns, you have accumulation. This is this line in gray in the upper plot. You have accumulation of a lot of conjugate of quercetin, which is flavonols. And then if you check instead legals that have very little, just a tiny portion that is UV absorbent, you have much lower levels of accumulation of these flavonols. And the same is true also when you compare, instead of different legals, you compare different parts of the same legal. So you can see here that the basal part of the legal accumulates this same quercetin glycosides, and they are accumulated at much lower level in the upper part that is UV reflecting. So this seems to suggest indeed that MIB111 controls this variation for floral UV patterns through its effect on accumulation of flavonoglycosides. In another plant species, that would be relatively straightforward to check. You could just take the allele that confers large UV patterns, put it, transform a plant that has small flower UV patterns, and see if you can recapitulate the original phenotype. So if you can get again plants that have large UV patterns. As many of you know in the audience, unfortunately, sunflower is not very transformation friendly. So we and other labs have tried for a very long time to come up with a reliable way of transforming sunflower and failed miserably so far. So we did the next best thing, which is going to Arabidopsis. So we look at the mid-boundary 11, so the homologue of the sunflower gene, which is expressed at low levels throughout the plant, but very strongly in petals. And indeed, when we went and checked Arabidopsis petals, we found that they absorb very strongly in the UV, which is a bit surprising because, as I said, usually UV patterns in petals are associated with attraction of pollinators, and sunflower is almost completely selfing plant. So we didn't expect really there would be accumulation of UV pigments, but there it is. And the other nice thing is that when you take a mutant for this gene, so a mid-boundary 11 mutant, accumulation of UV pigment in the petals is also almost completely gone. And that is also reflected when you look at the accumulation of flavonols. So you have different flavonols accumulating in the petals of Arabidopsis, but again, in green you have the wild type, so you have a lot of this. So these are chromatograms in the UV. So what you see is what absorbs in the UV in the extract of these petals. You see that most of the UV absorbing chemicals are flavonoids in Colombia, so in the wild type plant. In the mutant, all the accumulation of this compound is strongly reduced. And then what we did was we took the sunflower mid-boundary 11, put it into Arabidopsis, and what we found is that it can complement the mutant. So you have again petals that absorb very strongly in the UV, and you have again accumulation of these flavonols. And the same, so this is true when you use a constitutive promoter, so 35S, that would express this gene everywhere at very high levels, but also when we use the endogenous promoter from Arabidopsis, so we drive the sunflower mid-boundary 11 under the control of the Arabidopsis mid-boundary 11 promoter, we again see that you have accumulation of flavonol pigments in the UV absorbing pigment in the petals. The other thing to notice in this experiment is that, so we use two different versions of mid-boundary 11 from sunflower. We use a version from plants that have large UV patterns and small UV patterns, and that doesn't seem really to make much of a difference in how you can complement the mutant, suggesting that the difference between these two alleles is not in the coding sequence itself, so it's not the protein itself that function differently, but it's probably something more in how this the expression of this gene is regulated in different sunflower lines. So the next question is, how is expression of this gene in sunflower? Here on the first panel, you see RNA-seq data. This is published data, and we see that, as is the case in Arabidopsis, mid-boundary 11 is expressed at very high level in the legals and at low levels elsewhere. We look more in details, and we compare expression in the tip, in the UV reflecting part of a legal, and in the base, which is UV absorbing, the UV absorbing part of the legal, and we found that mid-boundary 11, as you probably would expect, is expressed at much higher level in the base, so where you have accumulation of the pigments. And this expression is particularly high in developing legals, so when the legals are small and they're just starting to get UV pigmentation and pigmentation in general, and decreases strongly in mature leaves. And this is consistent with a role in establishing these UV patterns early on in legal development. And we observe this pattern both in Annus and in Petiolaris, and we observe a similar pattern also for another gene, FLS1, which is one of the main genes that so it encodes for one of the main enzymes that controls accumulation of biosynthesis of flavonol glycosides. And this gene is also known to be directly regulated by mid-boundary 11 in Arabidopsis. And finally, we check a set of about a bit more than 40 different wild sunflower lines from a number of different populations, so from across the range. We just divided, so we selected lines that have large or small UV patterns, and we found that mid-boundary 11 is expressed at significantly higher level in plants that have larger UV patterns in their legals. So taken all together, this suggests again that the difference, so what controls the difference in UV patterns in wild sunflowers is the pattern of expression of different alleles of mid-boundary 11, rather than differences in the function of the gene itself. And this is consistent also with what we found when we sequenced this gene in a number of different wild sunflower lines. There's a lot of variation at this locus, as is the case for most of the sunflower genome, of course, across wild sunflowers. But there's no variation that is in the coding sequence of MIB-111 that is associated consistently with large or small UV patterns. While in the promoter region, we found, again, a lot of variation. There's a lot of structural variation. And in this case, instead, we found that there seems to be an association between the structural variants that are close to the SNP that has the strongest association in the GWAS analysis. So a lot of the structural variance seems to be associated with large or small UV patterns, meaning that it's probably, again, something in the promoter that changes the way this gene is regulated in different lines. As part of this experiment, we also sequenced MIB-111 from a few plants from other species, Petiolaris and Argophyllus. And in all cases, they seem to have the same allele at the promoter of MIB-111 as plants with small UV patterns. And so we went back to our SNP dataset and checked the genotype at the chromosome 15 SNP, so the SNP that had the strongest association in GWAS. We check what is the genotype at this SNP in different species, and we found that across different species, actually this tracks very well with the UV patterns in this plant. So you see here in this plot for wild Elianthus annos, the large and small allele for this SNP segregate at high frequency, and so you have this spread of values for legal UV patterns. In Petiolaris, Argophyllus, and Niveus, which are the species of wild sunflower, instead, the small allele is completely or almost completely fixed, and consistent with that, you have smaller floral UV patterns. While in cultivated sunflower, so this is the SAM population, the Sunflower Association mapping population that was mentioned before, the large allele instead is almost completely fixed, and consistent with that, you have mostly plants with intermediate to large floral UV patterns. So taken together, all these hopefully answers the first question. So how is variation for floral UV pattern achieved? This is through cis regulation at MIB-111 that affects the accumulation of flavonoid glycosides. And then, so we move on to the next question, which is why there is such variation? Is there any reason why there could be this variation in wild animals? As I said before, of course, the main suspect is pollinator attraction. So that's what usually is associated with UV patterns. So the function that is usually associated with UV patterns. So to look at this, we first did a very straightforward experiment. We took two lines, one with small UV patterns and one with very large UV patterns, put them out there in the field and check for pollinator visits. And we found that plants with larger UV patterns have significantly more visits from various pollinators. So this is like suggestive, but of course these are just two lines. They could be different for a number of other factors. So we have seen many of the possibilities in the talks in the past 2 days. So to try to compensate for other possible factors that might affect pollinator visits, we took a much larger sample of plants. So we checked pollinator visit in a set of 89 plants. These represent 49 different populations of wild sunflowers, so from across the whole range of wild sunflower, and they were part of a much larger field experiment, so a common garden with about 1,500 different wild annuals plants. And in these plants as well, we found that intermediate to large UV patterns tend to be preferred by pollinators. So they receive more visits, and in particular, the thing that stands out is that very short plants with very short UV patterns tend to be less attractive to pollinators. And this is consistent with what has been found also in other species. For example, this experiment in Rudbeckia, where they artificially alter the length of UV patterns by cutting and pasting petals, they found again that plants that had shorter UV patterns tend to be less attractive, while plants with large UV patterns tend to be more attractive to pollinators. So this is nice, but it really doesn't explain why there will be variation, right? So if there is one pattern that is better than the other, why don't you have just that pattern? And the thing that further complicates this is that variation across population of variation for UV patterns across population of wild Elianthus annos is not uniform. So you have a fairly clear geographic pattern with small UV patterns in the southeast of North America and larger UV patterns in central US and California. So this could be due to innate differences in pollinators, or it could be differences in pollinator community. It doesn't seem super likely though. So sunflowers, this is the native range of sunflower, of course, they have a number of different pollinators. So it seems unlikely that all of them would kind of coordinate to have different preferences in one area with respect to another. And also most of the major pollinators of sunflower are ubiquitous across North America. So while this doesn't exclude the fact that there could be effects of pollinators in driving this diversity, we thought it would be worth also looking at possible alternative explanations. And as I said before, flavonoid glycosides, so the pigments that are responsible for UV pigmentation in sunflower inflorescences, have been also associated to responses to a number of abiotic stresses, so abiotic factors. And we look in particular at 3 of them for different reasons, and these are UV radiation, temperature, and relative humidity. So for UV radiation is a kind of an obvious candidate for something that UV patterns in flowers would be important for, and this is because UV radiation is can be very detrimental to plant cells as it is to animal cells. And flavonoid glycoside, by absorbing this UV radiation, basically work as a sunscreen, right? So they would protect plant cells. And this is also supported by previous studies in Argentina and Serena. So they found there's variation for UV patterns in the flower of these plants. And what was found is that this variation, so plants that are exposed to that grow in places where there is higher level of UV radiation have larger UV patterns. So you have plants with larger UV patterns in closer to the more you get close to the equator. And the reason for that is believed to be because they protect pollen from excessive UV damage. So the flowers of Argentina and Serena are cup-shaped. And so the idea is that when you have large UV patterns, the UV radiation is not reflected into the pollen, but is instead absorbed. So this protects the pollen. And it has been found, in fact, that in the presence of UV radiation, if you have larger UV patterns, pollen viability is increased. So this seems like it could be a very good explanation for the diversity that we find. But the fact is that sunflower inflorescences are, of course, much more flat than the one of Argentina and Serena. And so it's unlikely that any significant amount of radiation would be reflected from the legals onto the pollen. And also, consistent with this, when we look for correlation between intensity of UV radiation and floral UV patterns across the range of sunflower, while the Helianthosanos, we found no correlation whatsoever. So it doesn't seem to be UV radiation that drives this diversity. The next thing we checked was temperature, and this is for 2 reasons. One is that we found a fairly strong correlation between temperature and floral UV pattern in natural populations of wild dahnios, with plants growing in colder places having larger UV patterns, UV absorbing patterns. So that's one reason. The other reason is the work that has been already mentioned several times in this conference. So the fact that UV inflorescence temperature is very important for pollinator attraction. So I won't go into the details of this because it has been explained many times, but wonderful work from the Harmer lab and Nikki's. And so we thought maybe naively that it could be, so you have larger UV patterns in colder places. So it could be that when you have more of this pigment, you have more UV radiation is absorbed, and this can help the flower warm up in colder places. The problem with that is, of course, that UV radiation is not very energetic. So it represents only between 3 and 7% of the total solar radiation at Earth level. And indeed, when we went and checked, so we use infrared cameras to look for flower inflorescence temperature. When we compare plants with small and large UV patterns, we saw no difference in the temperature of the legals. And this is true also if we check individual legals. We check the plants across multiple days from before sunrise until midday. And in all cases, there's no difference really in the temperature of the legals. You will see, however, that at least in these plants, there is a difference in the temperature of the disc. So plants that have smaller UV patterns. In this case, have quite hotter disks. Unfortunately, that has nothing to do with UV patterns in the legals. So if you remove the legals here in the last part of this figure, you can see that still the disk becomes hotter in these plants. The interesting thing is that when you remove the legal, you also remove the difference in pollinator attraction between these two lines. Unfortunately, that's just because no pollinator wants to have anything to do with some flowers that have no legals. So that's basically, there's no visits in plants that have no legals. So it's not temperature. And then the last thing we check is relative humidity. And the main reason for that is that across the about 2,000 different variables that we check, relative humidity is the one that had the strongest association with UV patterns across populations of wild sunflowers. So you have that plants that grow in places with lower humidity, so drier places, tend to have larger UV patterns. And this is interesting because, of course, relative humidity is one of the main drivers of transpiration, so water loss. And it is known that flowers, and especially big showy flowers like sunflower inflorescences, are a major source of transpiration and water loss in plants. So much so that it has been shown in different species that plants that grow in drier places tend to have smaller flowers to reduce transpiration. And the other thing that is interesting is that flavonoid are known to play a role in regulating transpiration. So if you take Arabidopsis plants and take transgenic lines that accumulate much higher levels of flavonoid, they will resist to drought much better. And this is because they lose water at a slower rate. So we thought it would be worth checking if that's the case also for sunflower legals. So we took 16 lines of wild sunflowers, pluck out a few legals from each of them and put them to dry and check how quickly they would dry. And we found that plants that had small UV patterns, so that had like lower level of accumulation of flavonoid dry up much faster, so lose water much faster than plants that have instead large UV patterns, so accumulate more flavonoid. This is not due to general differences between these lines. So we thought maybe the lines that have large UV patterns, they come from dry places. Maybe they are overall more adapted to have lower transpiration. That is not the case, because when we took leaves from the same plants and did the same experiment, we saw no difference between plants that had large or small UV patterns, meaning that it's not something that is a common characteristic of the whole plant, but it's really specific to legals. And this suggests that then these larger UV patterns might be important in drier places to reduce water loss, and so help plant conserve more water. Although, of course, transpiration is not always bad. Besides driving water transport within the plant, is also important to reduce damage from heat stress through evaporative cooling. So, very much like sweat in animals. And that might be the reason why you have these smaller patterns in the southeast of North America. In places where it's hot and humid, reducing water loss might not be quite as important as protecting petals against legals against heat stress. And indeed, we found that both temperature, so we found indication that both temperature and relative humidity are important could be important drivers of variation for this trait, because when we do the GA, so GA is pretty much like a GWAS, if you're not familiar. It stands for Genotype Environment Association. Just that instead of looking for phenotype of individuals, you look at environmental factors, so population level characteristic, and instead of looking at the genotype of individuals, you look at allelic frequencies within that population. But the general idea is the same. You look for correlation between these allelic frequencies and particular environmental factors. And we found that both for temperature and relative humidity, there are significant associations in the promoter of MIB-BRD 11. Again, suggesting that both of these environmental factors might be important in driving diversity at MIB-BRD 11, and as a consequence, diversity in floral UV patterns. So this hopefully this should conclude my talk. So the answer to the question why is there such variation, it's complicated. It's a combination of biotic and abiotic factor that probably drives this diversity. So We have found that pollinators are important, relative humidity and temperature are important. There's probably more factors that we haven't looked at that also play a role. And then, like, to conclude, I would just try to put together a few potential implications that may be more important for cultivated sunflower. So if there is something that we can learn from these experiments. And one thing is for sure that, you know, like, one needs to be mindful of trade-offs between, in general, but also between traits that are might be increasing pollinator attractiveness, because they could have other unwanted effect on biotic and abiotic resistance. Another example that comes to mind is floral scent. So it attracts pollinator, but it can also attract herbivores and pests. The other thing that maybe would be worth considering is whether, you know, the size of UV pattern in cultivated sunflower might be something that people want to look into to increase pollination rates in these plants. It's not clear to me that would be as important for cultivars. So one idea is of why these UV patterns are important. One is, of course, that they increase visibility and increase contrast between the UV absorbing and the UV reflecting part of the legals. But also, as you've seen, the disc is always UV absorbent. So, and the disc is where the pollen is, and the pollen is what mostly insects are interested in sunflowers. So the larger the area, it kind of simulates a larger abundance of pollen, right? And cultivated sunflower already have like such a large disc that maybe it doesn't make quite the same difference. And the other thing is that, as you've seen, cultivated sunflower, either by design or by accident, seems to be already fixed for the larger allele of MIB under 11. So this could be just a chance, or it could be that there has already been selection for plants, like known without knowing it, selection for plants that have large UV patterns so that they are more attractive to pollinators. There is still some variation within cultivated sunflower, quite a bit of variation. So one thing that it might be worth taking into consideration, and I am by any means a breeder, so I don't know if that makes any sense, but I was speaking briefly with Brent Halke a couple of years ago, and he thought it would be worth looking into that. Whether it would be important to have lines, so male and female lines for every production that have the same type of UV patterns, because insects are very good at learning learning color patterns in flowers, and they tend to prefer going to flowers that have a pattern that they're used to, right? So if you have males and females with very different UV patterns, maybe you wouldn't have quite as much movement between the two genotypes as you would want. With this, I conclude my talk. I want to thank a lot of people that have been involved in this project. So either in the UV part proper or in the larger effort to phenotype and genotype all those plants. And thank you for your attention. If you have any comment or question, I'd be very happy. So this is, there's a lot of expertise on sunflowers and pollinators at this conference. So I'd be happy to have any kind of question or comment later on. Thank you.